Hello, good afternoon. Yeah. Karen, if anyone needs to be admitted online, can you keep an eye on the admissions? Yeah. Okay, so thank you very much for coming to our seminar series. Yeah, I was just checked with Lindsay. Yeah, that she has the great chance to be the follow-up seminar presenter. She's following the director of the NIH that came and gave a great seminar to us on Monday. So no pressure. We know that you have the ambitions to be Fauci, take Fauci position. Yeah. But as you know, like we managed to put a fantastic international seminar series. Yeah, that we are having multiple. I think that we have 12 seminars between here and October. We put posters everywhere. And one thing that we identify is that it is a great place to have seminars because a lot of people come to Cape Town. Yeah. So Lindsay, unfortunately, she didn't fly all the way to the UK just for us. Yeah. So she's coming to be one of the presenters at the International Symposium of Microbial Evolution. Ecology, yeah, which is happening here in Cape Town at the ICC. Is starting on Sunday and go until end of next week. How many of you guys attending the ISME? Okay, so that's great, yeah, and then when she come, and then I say, Tulani, please approach her to give us a seminar if she can arrive a day before. And here she is. So I'm very excited to host her. Yeah, I promise not to do a long introductions but what I learned from her in the last 20 minutes. Yeah, I learned that she did her PhD in Cambridge at the Wellcome Institute, which, as you know, most of you know that we have a big relationship with them. Yeah. And then that after that, she went to Berlin, yeah, to work in Germany. And then she decided to go back to the UK, yeah. And one of the big objectives, it is to try to grow a global program that associated with microbiome, yeah. At the moment, she takes quite a high position. She's the chair of microbiome research at University of Birmingham, which is a very, very nice university. Yeah, not far from Oxford, but much more affordable probably than Oxford, and much nicer. And so what she's going to be present to us is the early life microbiota. So we're probably going to hear a lot about which microbiome the babies have. Is that right?
Yeah, we're going to be doing a bit of that. We're going to be looking at what mum's got as well, and we're looking at the gut, and also we're going to have a sprinkling of the skin microbiome as well.
Wow. So especially for you that have children or are planning to have, that may be a very enlightening talk. So thank you very much, Lindsay. And yeah, welcome. Yeah.
Perfect. Thank you very much. Yeah, so no pressure after speaking after the NIH director, right? So hopefully I'm going to make this a wee bit accessible, and apologies, I'm Scottish originally, so hopefully you can follow my Scottish accent, and I have a tendency to talk quite quickly. I will try and slow down, and hopefully if anyone's looking at it with blank faces, I can try and expand, but please do ask any questions at the end, and remember, no question is a stupid or a dumb question. So yeah, so I recently relocated back to the United Kingdom, to the University of Birmingham. I also have a group leader position at one of the UK government-funded institutes in Norwich called the Quadram Institute as well. And yeah, prior to that, I was in Germany, and I also did my postdoc actually over the IRC at the APC Microbiome Institute at University College Cork, which some of you probably heard about as well. So yeah, I guess I've travelled around a little bit. But what I'm going to try and do kind of for this, say for the next 45 minutes, is to give you a little bit of an insight into the work that we've been doing, predominantly looking at the early life developmental window, and kind of take you on a little journey. Some of the stuff, there's going to be, as always, be a lot of data. Hopefully you can kind of take it all in, but that's why you can ask any questions at the end. I kind of want to give you a bit more of a broader overview. But please just obviously check out kind of social media websites and stuff like that for any updates that we've got. Let's make sure perfect. And because I will talk too long, the most important thing for me to do at the very start is I'm the PI. So I sit at my computer, I try to answer emails, although I do get a lot, and I try to bring in funding for the fantastic people I work with, do the brilliant work that we do. So that's basically everything that I will be presenting. I've obviously basically done nothing. It's my fantastic team, our collaborators that facilitate the kind of the amazing science that we do. And I'm very privileged to lead and be part of these wonderful teams. Oh, and actually we've got one job advert just at the moment in terms of a postdoc position. We'll have another postdoc position, and there's probably another 2 PhD positions that will be coming up as well. So I'm also happy to chat about those roles if anybody is interested and fancies moving to Birmingham, where the weather is similar to this. Yeah, I know. That's what I was just about to say. It's just about to say. But Brum is great. So a little bit of introduction. Apologies for those of you that are very familiar with kind of this perinatal developmental window. So whenever we talk about kind of early life, we really talk about the first 1,000 days. And that's because they are critically important for the developing infant. And that's obviously because like the immune system, metabolic system, neurological systems, everything's basically getting developed, primed and matured within that 1,000 days. So it's a really, really important time for host development. And of course, the other thing that kind of co-develops across these 1,000 days is the microbiome. And that's also, these are the microbial communities that are both on the body, so kind of the skin microbiome, and obviously those microbial communities also reside within the body as well. But most of the niches that I'm going to talk about will be gut focused. So we are really, really interested in the team and kind of understanding what happens to the microbiome during this very dynamic time, understanding what the mechanisms are, but importantly, and hopefully I can convince you of this, if we can get to mechanism, we can probably develop the next generation of cost-effective therapies for at-risk populations. And that's what we have as our major goal in the team. So I've already mentioned our microbiomes co-develop with us after we're born. And in fact, you know, we're born sterile. So it's only through the process of childbirth and the first hours, days and weeks post-birth as we start to collect our hopefully beneficial microbiota. I'm going to talk about some of the pathogenic ones that might turn up, which we don't really want. But the thing that's really important to say is these microbes are critically important for programming our immune system, for obviously digesting the food that we eat. And they're also even really important potentially for cognitive and brain development as well. I'll briefly touch upon that because we've got a paper published this week on that. But the thing that's important to think about is we need to think about ecology because we're talking about communities, hence why I am session organizer and giving a talk on Monday at ISME. Because these are ecosystems and this is a developing ecosystem during early life. So this means that these microbial ecosystems are very, very sensitive to potential perturbation or changes. And some of the key kind of, I guess, external factors that might change or shift these early life microbiomes would be birth mode, so cesarean section versus a vaginal delivery, antibiotics. I'm going to give you an example of a group of babies that get lots of antibiotics and why that might be a problem, gestational age, so determining if you're born premature or if you're full term as well, and diet, because your microbes eat what we eat, so it's fundamentally important. So, and what I'm going to talk to you mostly about, as the first half of the talk, is the fact that premature babies, or preemies, are a group of, I guess, infants that unfortunately are potentially disrupted in terms of their microbiomes. So when I talk about preterm birth, this is kind of defined at babies, excuse me, that are born less than 37 weeks gestation. They're often very low birth weight, so less than 1.5 kilograms. And that represents about 1 in 9 live births globally. But I know from colleagues working in South Africa that actually the South African preterm birth rate is about 13%. So it's much higher than the global average as well. So there's big differences depending on where you are. But this rate is rising. Because these babies are born early, they're often, obviously the gut is kind of physiologically immature, and they also have an underdeveloped immune system because the immune system's already priming in utero, but preterm babies don't have that extra priming. Many premature babies are born by cesarean section. They get antibiotics. In the UK, if you're a premature baby less than 1.5 kilograms, you automatically get a course of benzoyl penicillin and gentamicin at birth, prophylactically, to potentially prevent nasty infections. And the prescription rates are obviously very, very high. So even though the healthcare professionals that work in these neonatal intensive care units, which is where these premature babies are kind of, you know, kept and cared for, they have a very different gut microbiota profile in comparison to their full-term counterparts. So they often have very high levels of potentially pathogenic microbes, and they're often missing some of key characteristic early life microbes, including bifidobacterium. And what we're going to start off by talking about is like when this goes bad in terms of pathogenic microbes, and then I'm going to hopefully, it will end nicely when we talk about beneficial microbes and how we can harness them as well. So, yep. So antibiotics plays a really, really big role, and I'll show you some data on that. But also because the gut is underdeveloped, it also means that it's not the same, I guess, environmental niche that facilitates colonization of potentially strict anaerobic bacteria, for example. And also the diet is often different in preterm babies as well. So it's multiple factors, which is why it's like so drastically different in preemies in comparison to full-term babies. And I think one of the key messages here is that, of course, the main role, not the main role, but one of the major roles that, you know, microbes play for us is colonization or infection resistance. And in the premature gut, that's basically completely absent because they don't have a normal microbiota. And that's really important for a particular clinical condition called necrotizing enterocolitis, which you may or may not have heard of. And we started working on this when I first got my first independent position. And honestly, I'd never heard of this, guys. And it was only when I started speaking to our consultant neonatologists and the midwives and the research nurses that actually I realized this was a huge problem. So NEC is one of the most, I guess, devastating gastrointestinal emergencies in neonatal intensive care units, affects between 5 to 15% of preterm births. And again, that range completely is different globally, depending where you are. Huge burden in terms of mortality, up to 40%. And if the babies do have NEC, and if they do get out the other end, there's serious long-term consequences, including neurological conditions as well. And importantly, NEC is linked to overgrowth of pathogenic microbes. It is multifactorial, but it's definitely linked to these microbes. And these are some of the clinical presentations. So these babies will go downhill very, very rapidly. This is why it's a big problem, because as in, it's not like there's a week-long run-up and you can treat them. They literally will go downhill within between 6 to 8 hours. It's very, very rapid. They'll present with this distended abdomen, which is normally gas buildup in the bowel wall called pneumocystis intestinalis. And this normally causes bowel necrosis. So this is where your neonatal surgeon will potentially come in, snip out the necrotic parts of the bowel, stitch them back together. But what you've done is you've just taken out a huge part of a bowel of a preterm baby, and you don't have that surface area for nutrient absorbent. So you can see all the other consequences that come from that as well. And importantly, from an infectious disease perspective, and this is why sitting down and actually chatting to the guys on the ground that are doing actually the clinical work is really important, is there's outbreaks of NEC. So they might not have NEC for 12 months and then the whole ward goes down, which would suggest an infectious agent. And one of the key ones here would be actually Clostridium perfringens, and that's just what I'm going to chat to you about here. Some of you may or may not know Clostridium perfringens. I remember this from my undergraduate microbiology degree in Glasgow. So it's a gram-positive and it's an anaerobe. It's basically ubiquitous, and it's known to secrete up to 20 different toxins. Thankfully, there's not a strain that has 20 toxins encoded, but they will have combinations of these toxins. It's one of the most common foodborne pathogens in Europe and in North America as well. But I guess from a very serious clinical condition, and this is kind of where at first, that's kind of why you hear it from a traditional microbiology perspective, is associated with tissue necrosis or gas gangrene. And importantly, and we do a lot of work in kind of the One Health sphere and work with kind of vet colleagues or people that are looking at animal health, perfringens is absolutely known as a causative agent for necrotic enteritis, say in chickens or in calves or even in horses. This is why it's always good to chat to people who are actually doing this as well. So where on earth is the link to premature necrotizing enterocolitis? Well, when you go back and have a look at the literature and even go back to the 1950s, interestingly, there's specific clinical cases and they're called gas gangrene of the bowel, which would suggest that there might be some symptoms that are very similar to what we see in the gut to what we see in tissues. And importantly, historically, perfringens has been linked to necrotizing enterocolitis, and that's kind of been done in animal models as well as kind of potential links with preterm babies. But it's only really because we've started doing longitudinal profiling of premature cohorts, doing microbiota profiling, that we've realized that perfringens might be a causative agent in preterm NEC. And actually, some of the seminal work that was done on this was by really fantastic collaborators down at Imperial College London, Simon Kroll and his team, like Alex and Kathy, where they showed that in a subset of their babies that develop NEC, they get this massive bloom of Clostridium and Clostridium perfringens just prior to diagnosis of NEC. And we were able to kind of work with Simon and his team to kind of develop this project. And of course, I say we, it's the royal we, because remember, I don't do anything, guys. I try and respond to emails and try and get money. This is where the fantastic Raymond Koo, who is a PhD student, who's now a senior postdoc in the team. This is Raymond had a really interesting kind of understanding the genomic underpinning of Clostridium perfringens and what might that mean for NEC. So this was a study that we had 5 different hospitals across the UK over a 5-year period. Fecal samples, and instead of just doing microbiome profiling, because there'd been quite a lot of work done on that, where actually we did the isolations of the bacteria whole genome sequencing, because of course that's kind of gold standard for doing genomic epidemiology potentially, and actually we've got some strains that we can actually do some experiments with as well. So this was work that was published just last year, so I won't go into too much detail, so if you want to knock yourself out on the 55 supplementary figures, feel free. But what I want to highlight here is that when we take all of our preterm baby poos and we look for Clostridium perfringens, a quarter of these poo samples will have perfringens, but a quarter of preterm babies don't get NEC. So why is the answer, is the question, and hopefully we came up with a bit of an answer. I think given the audience here, I probably don't need to introduce this so much in terms of kind of our phylogenetic tree that Raymond has put together. And this is just with our preterm isolates, but we did, and we, again, I did nothing. Raymond put this into the global context of perfringens. But what's important just to highlight here is that, and remember, this is going back to the toxins I mentioned, one of the toxins that perfringens is meant to ubiquitously encode and produce is PFOA, that's the gene, which is perfringilicin O. It's a pore-forming toxin. Interestingly, when we looked at the whole genome sequences of our preterm C. perfringens strains, and these were perfringens strains from healthy preterm babies and those babies that went on to develop NEC, what was interesting was that all of the babies that had developed NEC and had perfringens encoded this toxin, and all the preterms that did not get NEC had perfringens, but none of them encoded the toxin. So we thought that this toxin might be really important. The other thing we also found is that there's lots of other kind of virulence factors and colonization factors also encoded in the strains associated with NEC as well. So what the hypothesis was here was that we've actually got some hypervirulent perfringen strains that might be more causative for NEC, but we've also potentially got a commensal-like lineage or a hypovirulent Clostridium perfringens clade. Might not even be, I guess, not bad, might even be good for a preterm baby. So we're actually following that work up at the moment. And going back to some of those beautiful / horrible figures that I showed you in terms of the clinical presentation. So remember, this is the necrotic aspect of NEC. So when Raymond and Antia in the team went back and actually took our Clostridium perfringens strains and actually tested for does genotype equal phenotype, what they were able to show was that presence of the toxin in the genome correlated with really high necrotic capacity in vitro. So that's using kind of colonic cell lines as well as kind of small intestinal cell lines as well. And that basically correlated with presence of toxin as well. And the other thing that's really important to highlight here is, remember, I talked about outbreaks of NEC. So Clostridium perfringens, as well as being obviously ubiquitous, but part of the reason it's ubiquitous, it's a spore former. And of course, in a hospital environment, that's a disaster because these are environmentally resistant. They could be spreading around everywhere. So Raymond then went back and took a huge number of our strains and basically tried to find out what's the transmission potential of them. And is there a difference in transmission potential between the hypovirulent perfringens that are probably okay / maybe even good versus the ones that are associated with NEC. And initially, for those of you that actually bothered to read the slide, because I know I'm pressing very quickly, perfringens is anaerobic, and everyone says it's a strict anaerobe. I don't know how many of you work with anaerobic bacteria here. Most of them aren't strict anaerobes, right, guys? So in terms of vegetative cells, yes, they're not going to grow on the lab bench, but they're probably going to hang around and not physically die. And then if you put that back into an anaerobic environment, they'll basically take off again. And that's basically what Raymond and Antia were able to show. And actually the strains associated with NEC had a higher capacity to be able to basically start growing again as well. And then from a sporulation capacity, it was also clear that more of the, I guess, virulent associated perfringen strains had a higher ability to form these environmentally resistant spores than the perfringen strains that would be more like commensal or hypovirulent like. So this all links back to potential transmission as well. And actually, Raymond was able to do this within our data set because when he went back to have a look at the tree, we had a straight line. And a straight line means it's probably clonal. And this was really interesting. So we had about 30 strains, 1 SNP difference, but it was across 6 premature babies, across 2 hospitals, across a 100-day window. So there has to be transmission happening. So Raymond basically went back to the clinical metadata and did some digging. And basically what he was able to find was that there was an index case of this particular strain, so that was detected in an infant N18 in Hospital A. It was also detected in N17. Importantly, they're twins, by the way. We normally see perfringens in twins, which is interesting because it suggests that there's maybe something maternal maybe related there as well. Interestingly, both 17 and 18 had suspected NEC, and then they were basically chucked antibiotics, and in a good way, basically, they didn't develop NEC. But unfortunately, there was another baby on that ward that then caught that strain and actually succumbed from Clostridium perfringens NEC within about, it was in a 10-day window. And then importantly, the strain then moved to the sister hospital, because these are sister hospitals that basically transfer preterm babies depending what care that they need. So one of the babies basically went to the other hospital, took the strain with them, and then there was an outbreak at the other hospital as well. We didn't swab kind of parents. We didn't swab the surfaces of the neonatal intensive care like incubators or the personnel, because that's, well, the hospital lawyers were a little bit worried about that. But that's something I'd really like to do, because we don't know where it's coming from, and there's a lot of work to be done there. And then the other thing just to highlight as well is that just to confirm that we actually see this kind of NEC pathology in vivo associated with this particular toxin PFOA, was that we established a new murine model where we could orally gavage younger mice that have had a course of antibiotics to basically wipe out their colonization resistance or their microbiome. Interestingly, we don't actually see that the Clostridium perfringens persists in the mice, but it's mice. So we do use in vivo models, but they're 110% not perfect. So we didn't really see any interesting differences in colonization dynamics. We actually saw very little differences in overall microbiome profiles, but if you start to look at some of the other clinical parameters, including weight gain by the mice that got the toxin encoding perfringen strains from NEC babies had less weight gain, so suggesting that they're less healthy. And then when you open them up and look at their colon, you can see that the histology is much worse only in the mice that have the toxin positive Clostridium perfringens strains that have been infected. So even though the perfringens is gone, we can still see this direct damage, and we can also see really significant immune infiltration as well. So this is kind of showing as well that we get this hyper pro-inflammatory phenotype when the mice are dosed. So this is Clostridium perfringens, and well, I've said, they may be bad, but actually we think maybe there's some that might be quite good. But I think for us, it was important for understanding how a particular microbe that might be in the microbiome is really important for potentially a really serious clinical outcome in the most fragile of infant populations in premature babies. And then it would suggest also about the fact that can we do anything to actually improve the overall colonization resistance of our preterm babies so we can actually make sure that they're more healthy. So this is kind of where our other story comes in. So full-term healthy babies have a very resilient microbiome, normally dominated by bifidobacterium, and I'll tell you a bit more about BIF, who's one of our favorite microbes, and they have very strong colonization resistance. So then the question comes up here in a premature group is, can we actively supplement premature babies that are missing bifidobacterium with a probiotic containing BIF? And does that change the wider microbial ecosystem? And does that correlate to health outcomes as well? And this is bifidobacterium here, who's Y-shaped. I mean, like whatever rods or cocci, who cares? Ours is Y-shaped. It's like beautiful in an electron micrograph. And in fact, there's some really interesting information about why it's Y. So we're trying to do a little bit more work on this. So BIF, why do we care and why are we interested? So bifidobacterium is one of these kind of pioneer early life microbes that's really fundamental for overall health, both short and longer term as well. So it's found at incredibly high levels during this first 1,000 days, this key host developmental kind of window. So overlaps particularly with the immune priming window as well. High levels of bifidobacterium are associated with health, although defining health is basically impossible. So it does make our job quite challenging rather than going, you've got an infection or you don't. We're like, are you healthy or are you not? And everyone's like, well, what do you mean by that? It's very challenging, particularly from a therapeutic perspective. Importantly, and we'll come back to this point, bifidobacterium is found so dominant in the early life microbiota because it's evolved to be able to utilize specific components of the early life diet, so breast milk. and specifically the human milk oligosaccharides, these complex sugars that are found in maternal breast milk. So there's a beautiful evolutionary relationship between basically maternal milk, the baby, and the baby's microbiome, because these sugars actually pass undigested into the large intestine, and then they're actually digested by bifidobacterium, and then bifidobacterium produces the molecules that then feed the baby. So mum's milk doesn't feed our baby, it feeds our baby's bifidobacterium, and then baby's bif feeds them. So that's like amazing. So you get to some cool evolutionary stuff as well. And from a translational perspective, we're very, very lucky. So many strains and species have been used for many years as a nutritional supplement. So it means you can move quite quickly into the clinic. So they've got generally recognized as safe status, which is really attractive, although we're also trying to design our own targeted kind of next generation probiotics in the team as well. The problem is here is that bifidobacterium is one of the good guys, but it's exquisitely sensitive to some of the external factors I mentioned. So, for example, if you're a cesarean section born baby, you have less bifidobacterium because it's not vertically transmitted from mum to baby during childbirth. They're not antibiotic resistant, which is great, but not in a particular cohort. And again, if you're a formula-fed baby, you might still have bifidobacterium, but the levels will be much lower. So this is kind of where our Bambi study came in, and this was led by Christina, who's a PhD student in the lab, and Matthew, who's a postdoc in the lab. And again, this is a multi-center observational study. I won't go into too much detail because this was published a wee while ago, but just kind of bring through the story. We had some babies that were not given any probiotics. And then we had a group of babies that we sampled that we gave probiotics twice a day that contained bifidobacterium bifidum and lactobacillus acidophilus. And we sampled their poo samples the whole way during their stay in the neonatal intensive care unit, basically to see what does it do. Because most people give a probiotic and then they're like, that's it. Nobody does anything else. But it's as important to understand why it works. And if it doesn't work, why is it not working? That's all. The non-responders are almost, I would say, the most important group that we need to be looking at because we need to figure out something else for them. So the main, obviously, first question I mentioned about was looking at the microbiome. So this is just the first time point that we took, so kind of the first week after life in these baby poo samples. And this is obviously the preterm microbiome is incredibly low diversity, like crazy low diversity. Those of you working maybe with adults, preterm babies and even full-term babies are kind of much easier to kind of work with from a diversity and ecosystem perspective. But what we can see is that the supplemented babies had, unsurprisingly, higher levels of bifidobacterium and lactobacillus. We'll talk about what those species and strains are a little bit later. But importantly, they had lower levels of potentially pathogenic microbes that are associated with NEC and also late onset sepsis as well. So this is potentially good from an ecosystem perspective. And then if you look at all the other figures that we generated, we see this really stable profile. So higher BIF, low. Just to highlight here is that bifidobacterium levels in the supplemented babies were much higher across all the time points. Now, control babies, you can see. So a handful don't only have BIF at the first time point. They become colonized over time, but that's delayed. And that's really important because what you've probably happened here is that NEC, for example, happens in the first 2 weeks. So even though BIF is coming in the control babies, it's probably coming in too late. So that's why the active supplementation is probably really helpful. And I'll show you some clinical data about that as well. Importantly, everyone says about probiotics, you take them and you basically poo them out the other end and they don't colonize and they don't do anything. And I'd say that's probably true for the vast majority of probiotics that are on the market. But that's because you're probably a healthy adult and you've probably got resilient colonization resistance, and a probiotic actually finds it very difficult to engraft in the wider ecosystem. And actually, for our preterm babies, we were daily dosing them, but they stopped receiving probiotics when they were 34 weeks kind of corrected gestation. So as in what they would have been if they'd been born full term. So we stopped them. And then we were really interested to see is that if we stop the probiotic supplementation, does the probiotic stay or not, as the case may be? And interestingly, we found that it seemed to basically persist. So we wanted to look at that in much more detail. And again, well, I wanted to look at it, but again, of course, I didn't work. Christina did all this work. And then what Christina was able to show was that the supplemented babies were absolutely dominated by specific species of Bifidobacterium bifidum, which is the probiotic species. And our control babies that did get BIF, they weren't colonized by Bifidobacterium bifidum. They were colonized by Bifidobacterium longum, subspecies longum, Bifidobacterium brevi, for example. So quite different Bifidobacterium. And actually going back to poo samples after the supplementation had stopped, and Christina did culturing for Bifidobacterium, she isolated strains, she did whole genome sequencing, and then she lined those genomes back up with the genome sequence of the probiotic strain we gave, they're the same. So what we can show there is that even if you stop the supplementation, the probiotic sticks around, and it sticks around for a long time as well. And in fact, we got data from 2 years of age and it's still there, which is amazing. So the question is, why does it stick around? And why do we, like, what's the particular underlying mechanisms behind that? So going back to diet, and Bifidobacterium's favorite food of all time being human milk oligosaccharides. So for our particular study, all of the premature babies either had expressed breast milk from mum, because of course very preterm babies can't breastfeed, obviously. So you get expressed breast milk, my mum kind of nasogastric tube, or they'll get donor breast milk, and donor breast milk banks are fab. So none of our babies were basically on formula. So that's really important to say. And then when you look at, and we strategically did think about the probiotic we wanted to give. The Bifidobacterium bifidum strain we used has the enzymatic capacity to digest human milk oligosaccharides. So even though I say Bif's favorite food is HMOs, not all strains have the enzymes that can degrade HMOs, or they might be able to degrade a couple of different types of HMOs, but not all of them. So again, it's thinking much more particularly about things. When you grow the probiotic strain in whole breast milk, it loves it. It grows really, really high, which is fantastic to see. And then if you give it specific human milk oligosaccharides, so purified human milk oligosaccharides, they also grow. And that basically links back to the fact that they've got the genetic potential in their genomes and that they're actually able to utilize those HMOs as well. So they're potentially metabolizing or using this early life diet. What does that mean? So using metabolomics, which is a really nice way of kind of taking the next step to kind of functional profiles in the microbiome rather than just looking at taxonomic profiles. This was a great collaboration with John down at the University of Southampton. She's just an NMR plot here. What we can see here is that our BIF and lactose supplemented babies versus our control babies, they've got quite different NMR spectra, right? And what does that actually mean? Well, importantly, our supplemented babies have much higher levels of short-chain fatty acids, including acetate and lactate. And actually, Christina went back to the preterm baby poo samples, so the supplemented babies and the control babies, and she pH'd them like literally like basic science. And importantly, the preterm sample, the preterm babies that got the supplement, the probiotic, had a pH, fecal pH of 5.5, control babies pH 7. That is a huge difference on a pH logarithmic scale. And that's really important because a lot of pathogenic bacteria don't like those lower pHs. So just by probably changing the physical environment of the preterm gut, that's probably one of the reasons why we're seeing these whole scale changes. And actually, when you correlate this all up, so you correlate your taxonomic profiles with your metabolite profiles, including acetate and lactate, Bifidobacterium is associated with higher acetate and lactate and lower levels of human milk oligosaccharides. So basically, the control babies have got HMOs in their stool sample because they poo them out because there's no Bifidobacterium. And the supplemented babies don't have any HMOs there because they're used, metabolized, and then the acetate and lactate are produced as the end point of the fermentation pathway. And that basically means an acetate and lactate are acetic and lactic acid. They're acidic compounds, they're short-chain fatty acids, so they reduce the pH. So after doing a whopping huge clinical study, it's the first clinical study we ever did, and we learned a lot because it was on a wing and a prayer, came back to pH after all that, but that's kind of cool. But our clinical colleagues were like, we don't care that you changed the microbial ecosystem, even though we cared because it was kind of cool. What does it mean from a clinical perspective? So Claire was a trainee clinician that was with us in the lab, and basically she went back to see what this means from a clinical perspective. And actually, if you look at babies that received the probiotic versus those that did not, there's a 50% reduction in necrotizing enterocolitis, and actually a 50% reduction in all cases of mortality, which is pretty amazing by just giving a BIF that reduces the pH. So that's kind of like amazing. It's incredibly cost effective. So this costs the NHS probably about £30 a day. So it's really, really good. And this is kind of what we want. And the impact of this, which is kind of something that's always really important, certainly for me and for the team as well, about kind of what this means. And part of our work now has been cited in the World Health Organization care guidelines for preterm babies, which is like super amazing. And we've had other units across the UK start to use probiotics and also across the world as well. So I get random emails from people going, we want to do this, what should we do? And it's really amazing to be able to potentially help them set up as well. And just to clarify, I'm not a clinician. I'm only a lowly scientist microbiologist, but we can also have impact by working with bigger teams as well. And that's been really amazing. And at the moment, Paul and Claire are updating this for the next 5 years of data. So based on this, we had Charlotta, who was a PhD student at the University of Graz when I was at TU München, and Christina, who's her supervisor, which some of you may know. Christina, she actually works in Archaea, but they were doing a preterm study as well. So Charlotta came and spent 6 months with us in Germany, and they were really interested because the clinical care of preterm babies in Austria is literally ad hoc. So each unit does a different thing. And it was really important to understand what thing was going to be the best for the preterm babies. So Charlotta basically obviously kind of and reassuringly kind of reproduced a lot of the data that we and other fantastic teams have been showing in this area. But importantly, what she was able to show as well was that the probiotic supplementation in one particular unit wasn't just associated with kind of differences in taxonomically, there was a reductions in multi-drug resistant bacteria in carriage. And that's obviously really important from a sepsis perspective and using antibiotics in an appropriate way. So that's preterm babies. So preterm babies come out early. And then at the end of that study, because I was really interested to understand, well, can we do something even earlier to potentially mitigate against even preterm birth completely? And this is important because obviously during, in utero development in the womb, the placenta, the maternal gut microbiome has a potential to massively influence the types of energy molecules and other signaling molecules that are given to the baby during birth. To clarify, there is no placental microbiome. That's probably low biomass samples, not appropriate controls. But absolutely, in terms of molecules and other things that are going to be passed over, there is. Or if there is something in the placenta, unfortunately, it's probably going to be associated with a negative health outcome because it's an infection, for example. And what's important here is that at late gestation, there's lots and lots of kind of nutrients being transported because there's lots of kind of developmental and growth happening in the fetus. And what's really interesting is our good friend and colleague Omri published this work a couple of years ago, where they looked in mice as well as humans. And what you see is a steady rise of bifidobacterium across pregnancy, which is really cool. And actually, they linked it back to progesterone. So BIF responds to progesterone. So that's wild. So again, thinking about how the whole just pregnancy itself, what's happening, can change mum's microbiome. So basically, she sets up for helping the baby and then post-birth as well. So there's like so many other like amazing things to be doing here. And I won't go into this in too much detail, but I just want to highlight some more of the mechanistic stuff. This is obviously stuff that we've done with fantastic collaborators, including Jorge and Amanda at the University of Cambridge at the Trophoblast Research Centre, and Zoe, who's a brilliant postdoc in the team. And this is in mouse models, because of course you can probably, ethically, this is not something that you can do, certainly not straight away in humans. But interestingly, using germ-free mouse models, these are mice that have no microbes at all, and specific pathogen-free mouse models, what we were able to show is that just supplementation of Bifidobacterium brevi, in this case to the female mice during pregnancy, changed placental kind of morphology, increased nutrient transporters, including those for acetate, for example. And we saw transcriptional responses at the fetal liver and fetal intestine level, showing that there was active things happening just because we gave Bifidobacterium as well. So that's really amazing. And I've currently got grant in that's going to hopefully try and do this in humans. We'll see where it goes. I'm really hoping that everyone's excited about this as I am. Probably not, because I'm overly excited about everything, but anyway, it's fine. And then most recently, Jorge actually published another fantastic paper in molecular metabolism. Although just to tell you, I think that's now been taken down because we're now going to do a press release, where we don't just see that maternal supplementation of Bifidobacterium in mice is important for that placental or fetal programming in terms of the liver and the intestine. There's also changes in brain metabolism and brain development as well. So that's like super amazing as well. So that's that nice mechanistic underpinning about why BIF is potentially really important in different windows as well. So what's next? So the Bambi, the preterm study nearly killed me. So then I thought, well, what will we do next? Let's do another clinical study. So this is Pearl. So Pearl is a pregnancy and infancy cohort that we established in Norfolk in the UK, and which obviously has been unsurprisingly disrupted somewhat by COVID. It's a observational prospective study of 250 pregnant women and their newborns up to 2 years of age. We're taking stool, vaginal swabs, skin, milk, blood, urine, dietary questionnaires, lifestyle questionnaires, and all primary and secondary outcomes. I'm going to show you a little bit of data from this. This is very much ongoing. We're doing deep shotgun metagenomic on all of the samples, and we're doing culturing for microbes so we can actually think about therapies and doing experiments. And we're also planning to do metabolomics and maybe even metaproteomics as well, and to put these into different in vitro systems. And we got our final sample, well, technically our final samples in April, but we've got a handful of samples that are coming through now for August, because we've actually got 262 women on the study as well. So if any of you are thinking of, and I know there's a couple in the audience, of thinking of setting up kind of a new pregnancy or life study, we published the protocol. Very happy to share SOPs and basically knowledge, because this was a lot of work that basically Sarah Phillips, who was kind of the coordinator for this, did a huge amount of work to basically get this up and running. And we've tweaked it, and I think it's a really effective way to really kind of get these samples really effectively and to do this type of work, and retention on a longitudinal study as well. But this is where we went beyond the gut, and that's because I wanted to initially do skin swabbing, so taking basically just a swab and just swabbing it on the forearms for mums and babies, because I was really interested in how skin microbes might influence gut microbes in cesarean section born babies, because that was when we established the study, that was what was interesting. And then obviously stuff has happened since then. And we now realize that obviously skin microbiome, just for the skin microbiome, is a really important kind of data set as well, because skin microbes also play a fundamentally important role for kind of maturing and developing the skin immune system, for example. And they're also correlated very strongly with healthy skin, or if these communities are perturbed, maybe with skin associated conditions as well, like including atopic dermatitis. But what's important is a lot of the skin studies that have happened so far have focused on adults rather than infants. And actually the microbial community in infancy is quite different than adulthood as well. And it's linked to early life outcomes. And actually Ileana, who's the PhD student, who's actually will be submitting at the end of September, did a really nice review about kind of where we are with the skin microbiome. And there's actually a lot of kind of questions still to be answered. And Ileana's been doing that as part of her PhD. So very much watch this space. But one thing we realized, and probably for all of you here, again, I'm talking to an audience that will be well aware of this, the skin microbiome is more challenging to work with because it's lower biomass. So even though the stool is, as I said, low diversity, there's still a lot of bacteria in there, the skin can be much more difficult to work with. And nearly all of the studies previously done were done on 16S rRNA amplicon sequencing, and that's partly because low biomass, and obviously kind of it's cheaper as well. But actually, if we want to get down to strain level and functional information, we really do shotgun. And in fact, we probably don't just want to do Illumina short, we probably want to do long read shotgun as well. So Ileana spent a long time optimizing, making sure we get enough DNA from our swab samples to be able to look at this in detail. And she published this in Microbial Genomics just last year. And I'm a senior editor for Microbial Genomics, publishing for the community. Send me all your genomics and microbiome papers. And that okay, so that's my pitch done. But it's for any of you that are working not just with skin, but maybe other lower biomass samples, please have a look at Ileana. She did a fantastic job at kind of really doing that. And I think what's important to say here is that we did this on adult volunteers as well as some of our pearl samples that I mentioned previously. What Ileana was able to do is you can snip the swab in half. Half goes for shotgun metagenomics, the other half goes for culturing. So we've got strains that we can play with as well. So it's a really efficient way of getting really fantastic work. So just last kind of couple of slides here, because this is very much a work in progress. So it's following the skin, but vaginal and gut as well. Ileana, as part of her PhD, couldn't wait to the end to get all of the samples together because that's not how it works. So she's taken a subset of our Pearl cohort where we've got samples from trimester 1, trimester 2, trimester 3, birth, and then all the way up to 2 years of age, and a group of infants as well. And she's got shotgun metagenomics sequencing on skin, vaginal, and gut. And we can see, unsurprisingly, we can see some actually dynamic changes. And apologies, I think the resolution isn't great. I normally like to say, if anyone's got a bug that they see on here that's interesting, we've probably got cultures for it. But yeah, you can maybe comment. I'll maybe give you another overview afterwards. And just some very kind of preliminary data that Ileana's been putting together here is that, interestingly, and we see what we see on the skin, both in pregnancy and in early life, is kind of what others have shown, but we've been able to do that at better resolution. And I said Ileana's doing some strain tracking work at the moment, which is thrilling. So we've got Staphylococcus, Streptococcus, Micrococcus, Acinetobacter, and Moraxella. And I said we've got cultures for those as well. So we see some quite dynamic changes across pregnancy. Does this link to hormone changes? To be decided. So we need to find that out still as well. And we also see quite kind of, I guess, transient and dynamic changes happening in the infant skin microbiome. And I think one question why you always need to do longitudinal studies is if you take one time point, is that a colonizer or is it just there and you're not going to find it tomorrow? So if you can do long-term, particularly strain tracking, you can confirm that something's there and it's actually a core member of the skin microbiome if you follow it longitudinally as well. And part of the work that Ileana's doing with kind of the help of Raymond is she's been assembled metagenome assembled genomes as well as our whole genome sequences as well. We've got over 4,500 mags. God, I love these trees. So this is very much a work in progress, but again, we can actually track that dynamic strain kind of changes over time and link it back to, was the baby born cesarean section? His mum had antibiotics. We've got all of that clinical data that we can really start to tease apart those differences. And we've also got access to some really cool skin models as well, in vitro models. So we can actually confirm, is it a stable colonizer or not? Let's chuck it on skin and see what happens as well. Because we don't just collect data, we like to do some like experiments with it as well. Again, I don't do anything. The team are fantastic. Work in progress, but watch this space. And unashamedly, for all of you, and this is miles ahead of time, this is 2026, and I know we've got ISME that's starting on Sunday, but please have a look. We've got Microbiome Interactions Health Disease is the last one that I'm on the organising committee for. There are travel bursaries available, so sign up for that because it's always a fantastic meeting as well. It's at Cambridge at Hingston Hall, so beside Sanger. And just one other thing, the other thing I feel passionately about, and I always like to highlight this, is we're scientists. We have a responsibility to communicate what we do to everybody else, particularly for inspiring the next generation. So we've established something called Guardians of the Gut. So we built a giant walk-through gut. For those of you that, I don't know, watch UK TV, this is Brian Cox, who's a very big science communicator. So he wandered around our gut. So my team are not just fantastic scientists. They're artists, builders, and engineers, and electricians. 5,000 LED lights. You can walk physically into the gut. You press buttons. So if you're a C-section baby or vaginal delivery baby, the colours change depending what your microbial profile looks like. So we've taken that to the Royal Society Summer Exhibition. We've taken it to music festivals as well. So that's been really, really cool. But the other thing on the back of that is that we've set up a freely available primary school lesson pack for kids to learn about their microbes, why they're important, and why feeding them and looking after them is really important as well. Because science, particularly for the UK, a little bit of primary school, but there's not that much. And a lot of primary school teachers don't necessarily come from STEM backgrounds as well, so they don't feel comfortable or confident teaching science. So they can either teach the lessons themselves or they can download a video of me giving a lesson to the kids as well. And that's been, we've had hundreds and hundreds of schools sign up. We ask for their knowledge before the kids come into the session. Then we ask afterwards and 6 months later to see if they retain the knowledge as well. Because again, if you're doing any engagement stuff, just like science, you need to act, does it change things, right? You need to actually have that, qualitative and quantitative data. So we unfortunately weren't able to do very much in Germany, and we're basically going to kickstart stuff back in the UK as well. And I've been chatting to colleagues in different parts of the world about basically doing a country or regional specific version of Guardians of the Gut, because we've got all the materials, and if it's a case of potentially tweaking it or changing it, changing language, changing videos, then it's much more easier and accessible because we've done a bit of the work behind there. And then that's it. And thank you very much for your attention.
Thank you very much. I'm so glad that the director of the NIH was not here, otherwise she would be ashamed that you did better than her. Yeah, as you know very well, one of the big characteristics of this symposium is that after great presentations like that, we also like to grill the presenter and ask multiple questions. Yeah. So please, who wants to go first? Sorry, we like to make you work hard for.
Yes, thank you very much. It was really a wonderful overview. I learned a lot. So I'm a medical microbiologist. I used to work in the Netherlands. Intensive care was always so difficult for infection prevention. Like so many times, all the kids had the same bacteria, and after many audits, it didn't really improve much. I was wondering the association you saw with Clostridium with the toxin and the occurrence of NEC. Is that also, could that be used as like a screening, that if kids have this, that you maybe put them in really, because the care is usually an island setting, and maybe these kids should be really separate?
No, absolutely. And I think this was something when we worked, well, I said we were also chatting to the microbiologists as well, the clinical microbiologists and the teams. And we realized that this is maybe something exactly that you could potentially be coming in, because I know that they obviously look for, certainly for adult ICU, they normally obviously look for particular like E. coli or whatever in terms of what they might have. And then they might be, like, as you say, put into separate wards. For preterm babies, it's a little bit different because they're kind of born and then they're kind of like rushed in. But if we did do that, it has to be really quick. Because for example, if it's a spore form or whatever else, it's potentially going to get around a ward really quickly. So I think one thing here, for example, some of the other work that we did a couple of years ago that we're still working up was actually using like the MinION to potentially do really quick, rapid diagnostics to be like, what's there, what's the antibiotic resistance profile as well, and what will that mean in terms of outcome for the babies. So we're still kind of working that up at the moment. But I think there's very specific challenges with that group in that setting as well. And I think we need to be a bit more kind of innovative with thinking about how we do that. And just to highlight, perfringens is definitely a problem. We think it's probably associated with the worst type of NEC, but Klebsiella is really important for NEC as well, Enterobacter. So there's probably a range of microbes there that we probably need to be looking for example, and that's probably why a metagenomic rather than targeted diagnostics is probably more effective as well. But yeah, we are trying to have a look at that as well, but I appreciate that's a really difficult setting to work in.
Yeah. And so when you saw Clostridium perfringens taking over, is there something that triggered that, like antibiotics, or was there any factor?
Yeah, it's a really good point. So one thing I mentioned is Clostridium perfringens is incredibly quick growing. So I don't know if anyone works with E. coli and everyone's like, oh, E. coli is really fast. Clostridium perfringens doubling time is about 7 to 8 minutes. So it wins. So you can imagine if it gets in, it goes. And the other thing it does, it produces hydrogen sulfide gas as well. And that's one of the reasons probably why there's direct pulp, like as in, so the toxin's really important, but there's actually kind of like holes probably through gas buildup and other necrosis as well. So antibiotics are potentially important there as well. What we find is those babies have basically no BIF at all, even very low levels of bifidobacterium. And those babies are normally the ones that are the most born the most prematurely as well. So they do have some other clinical challenges as well. But it kind of looks like whoever gets in first wins, which is why I think you see perfringens in one group of premies, but in that same cohort, we saw like another group that had Kleb in another ward. So as you say, probably it's in and then it just gets spread around. So I think probably there may or may not be something specific there. I think it's maybe just what it gets when it gets in, and then it's just basically able to grow and replicate.
Thanks.
Thank you for your talk. It was very interesting. I have a question with regards to breastfed babies. So how does the mom's diet, if a mom changes her diet, how does that impact the microbiome, the developing microbiome of the infant?
So great question. And actually, that's one of the questions we're hoping to answer with Pearl, because one of the things that's very difficult to do for any longitudinal study is collect good dietary information, because most people just can't be bothered to do it, or they just lie. So that can be very challenging to do. So there's definitely some work there suggesting that, for example, like, you know, if mum's having a very high fiber diet, actually that potentially even influences what microbes might be found in her breast milk, that then influences, because there's also breast milk microbes that might also influence seeding of the baby in longer term. There's also some suggestions that human milk oligosaccharide composition can be also changed by diet. But again, what the underlying mechanisms are, I don't think we know, because a lot of this is just association studies, and we don't really know what the underlying phenotype is. But absolutely, there will be a huge role to play. And like, for example, also like preterm birth as well, there's definitely links with like undernutrition or overnutrition potentially might be linked to preterm birth, which might be linked to differences in like, you know, gut microbiome, vaginal microbiome as well. But that's why we need to do more of these longitudinal studies, try and get as much data, but not too much data that everyone's like, I'm not taking part in that study because it's ridiculous. very fine balancing act to do. But no, it's a great question. I think still we need to do quite a bit of work in that area.
It's probably especially important for Yashni, because I believe that you just had a baby. Yeah. Are you keeping a food journal? Yes. And do you see any much difference in how the baby? Yes. Okay. Thank you. Yeah, we had a question here. Think about yours.
Actually, my question is quite similar. I mean, you mentioned that the twins generally have Pfiffingens, and that there might be a maternal connection, but you did also speak quite a bit about what that might be. And so I don't know if you need to answer.
No, twins are interesting. So obviously for a preterm cohort, just by definition, we get much more like twins and triplets because they're preterm, because there's extra babies in there and they're normally born early. And that might also be the fact that there has to be like a cesarean section for obviously health of the baby, but also health of the mum as well. So interestingly, we do see potentially, for example, Pfiffingens that we see similar, but we see many, many twins that have some similar microbiome profiles, but more often than not, we see quite drastically different microbiome profiles between twins. But when you go back to look at the clinical care, it's probably because one of the twins has had antibiotics, the other one is not. And in fact, the role of host genetics in microbiomes plays a very, very limited role in comparison to all the other external factors like diet, drugs, for example. The only really strong link up with kind of, I guess, genetics, the microbiome, is Bifidobacterium. But it's actually those individuals that genetically should be predisposed to lactose intolerance, but they're not symptomatic. And when you go and look in their gut, it's because they've got a lot of Bifidobacterium, and Bifidobacterium's favorite, simpler sugar is lactose. So there's a beautiful, in terms of BIF basically wipes up the lactose, so then you're not intolerant, even though you don't have the enzyme yourself because of the SNP to be able to do it. So, but we need to do much more kind of work there as well. We do actually have approval to do, if we wanted to go back and actually do get genomes for mums and the babies, the genetics. But well, one, I'm gonna have to get a lot more money to be able to do that, and then try and link that all up. Yeah.
Yeah. I have two questions. Now it comes from the microbiome people. So
excellent.
Let's see if they believe your presentation. Yeah.
Thanks for the great presentation. You mentioned that beef does not generally have resistance genes. So does then prophylactic antibiotic therapy make the situation worse in kids who already have low beef balance?
Yeah, and it's a really good question. That's basically the problem. So we give antibiotics and we're hopefully going to get rid of the pathogenic microbes, but data that I didn't actually show you from Bambi, if you look at antibiotic usage and then you look at like the taxonomic profiles. So if you look at antibiotic usage and then BIF, it's significantly decreased. If you look at antibiotic usage in Klebsiella, E. coli, Enterobacter, they're not reduced because they're nearly all multi-drug resistant. So even though the antibiotics are meant to be there prophylactically to prevent infection, probably what's happened there is they basically reduce colonization resistance and let the multi-drug resistant pathogens increase. So actually Raymond is just finishing off a big study, and this is a deep shotgun metagenomic data set for probiotic and non-probiotic supplemented premature babies with antibiotics with not, and trying to find preterm babies that have not had antibiotics is really, really difficult. And we'll hope preprint maybe next month where we can actually show that in much better detail. And that might be something in terms of clinical care guidelines we need to have a look at. But from speaking of neonatal colleagues, it's really challenging, right? Because these babies are much higher risk of infection. And then you're telling them, don't give antibiotics because it's going to be bad. And then for the couple of babies that you don't do that, what will happen? Even though you have all the rest of them, will probably actually be much better and much healthier. That's a really, really difficult kind of call and decision to make. And there'll be probably much bigger studies for evidence-based required to do that. I will, for those of you that are coming to ISME, and apologies, you are going to see some of the same stuff, but I am going to talk about antibiotic resistant BIF and sometimes when it is there and in what populations as well.
Hi, thank you.
Just stand a little bit. Yeah.
Yeah, I think it's actually okay.
Yeah.
So I'm just going to get to my notes in a moment, sorry, because there's a couple of questions. The first question, the Clostridium perfringens study. So my question is that, did you see a different profile to the neck from the neck-related strains that were for the pre-terms versus the normal terms? So you mentioned that there is a difference between the commensal strains, and there is also the neck-related strains. So were there any different profiles for the preterm babies versus the normal birth? Yeah.
Yeah, no, so that particular study, we only looked at preterm cohorts. However, we have looked in full term cohorts for perfringens, and for perfringens does appear to be common. And it does look like that they're probably lacking the toxin as well. So we've actually been in discussion with collaborators actually over in the US that have got like a piglet model for potentially actually understanding the, well, one, they're really interested from the toxin and neck, and a piglet model is a very good, much better translational model for preterm neck. I'm really interested in the ones that don't have the toxin, the commensal strains, and I want them to take extra samples to see if that's actually influencing epithelial barrier integrity and immune system development. So I think hopefully we'll have a little bit more information on that. But it does seem that, again, it's not, it's not kind of one or the other. Again, we need to understand that. But that's because when we started looking in the databases for perfringens, and we were like, oh, this toxin is like it's a part of the core genome. All of the strains, nearly all the strains, 96% of the strains in the genomes are all from infection-associated conditions. So everyone thinks it's a pathogen because nobody bothers to look at healthies and what the microbes are doing there. That's why those studies are really, really important as well, to understand what we take as this is really important versus like actually in normal people or normal babies or babies without overt disease, maybe actually these are good and we maybe don't just want to get rid of them all either because they might be a really important role.
Okay, and on the next study as well, you mentioned that you did histopathology in mice. What type of mice did you use?
So that was C57 black 6 mice, and they were, let me get it, 3 weeks old.
Okay, were they female or male?
Female.
Okay, and do you think that if you were to use male mice, would see different type of?
This is thank you for bringing that up, because that's really, really important. And because we normally, particularly for in vivo studies, we will normally obviously only look at particular like sexes, and that partly because that normally works with the phenotype that you're looking at. I'm saying actually scrap that. It was male and female mice because it's pups. So actually we had both of them together. So that was me showing me forgetting there. But normally we would do, so when we did the older mice, it's always females. Pragmatically, it's because you can put them in a cage and they don't kill each other like the males do.
Okay. And do you think that the histopathology that you observed for the colon would be the same with the human babies?
So we were hoping to get some of the histopathology that we saw, so in terms of like direct damage and this big immune infiltration. So when we spoke to the like histopathologists at the hospital, the pathologists, we can see some of those traits are very similar, but they're not completely the same. But that's because even though we tried to do our in vivo model, it was probably quite a mild form of neck in comparison to what we'd seen a preterm baby, which is why the potential new collaboration with colleagues over in the US with a more translationally relevant model, we might be able to look at that and it's much more specific because again, it's a human perfringen strains in a mouse. So caveat with that, great to kind of show it a little bit further forward, but it's not perfect. And yeah, absolutely.
Yeah, but okay, I will just try and summarize the Pearl study. That was a huge study, 250 participants, and you also made a couple of sample types. Did you sample blood as well?
So we've got cord blood and we've got some blood from the women if they're getting taken bloods routinely. And we've also potentially got bloods from the babies as well. But our blood samples are not as complete as our other sample sets because they're invasive. And when we did our patient public kind of, engagement and we were chatting to individuals before we started the study, blood is one of those things that if you say that's mandatory, if you sign up, we want to take blood, they'll all turn around and go, no. Yeah. So again, we needed to, that's why we did all of the kind of background chatting, making sure what we were doing was realistic. So we do have blood samples. It'd be really interesting to have a look at them. Yeah. But unfortunately, it's not, we don't have blood samples from all the different time points from all the participants, just because we said that they were like, I said we would take them as part of routine care, and they were kind of like, you know, if you want to give them, it's fine. They weren't mandatory.
Yeah. So you haven't looked then at the host responses.
We've got PBMCs in liquid nitrogen.
Okay.
And we haven't done that yet because we can't. This is why I normally show Pearl, because we've got some specific stuff we want to do. We're not experts on everything. It's an incredible resource, and we are always looking for people that might have really specific expertise or questions that they can bring to the table and that potentially we could work with, because the whole point of this is that this will be for a wider resource as well. So yeah, happy to kind of chat further if that's of interest, because we've literally got them and I'm like, we need to do something.
That would be really interesting to correlate the host and the pathogen data.
Yeah, absolutely.
Thank you.
Thanks.
Yeah. And don't forget to apply to the conference. Yeah. March is at the Sanger, is a beautiful place. Yeah. And maybe we even like Birmingham with the weather. Yeah.
Sorry, I don't know. Okay, thank you very much for your presentation. Okay, I have two questions. So the first one is on... Okay, can you hear me now? Yeah, okay. So the first one is, I think you talked about something about lactate.
Yeah.
Yeah, so, well, I have a basic understanding of the microbiome in TB. So short-chain fatty acids, butyrate, acetic acid, and lactate, like, they cause a lot of problems in TB. Do you think they have like an impact in babies? Like, are they important?
Yeah, so in the microbiome field, the dogma is short chain fatty acids are like really good and really important. And certainly for butyrate and probably propionate, that's kind of true, although there's some interesting data saying that if you've got a lot of butyrate, actually it's probably not a good thing. Acetate, there's some beautiful work showing that acetate, programs like the immune system, and it's also used by lots of other microbes in terms of cross-feeding as well. Lactate is an interesting one. So I've got colleagues that are working on this in terms of the microbiome, and there's obviously lactate utilizing bacteria. And something that we haven't done yet is actually seeing about, we think lactate is probably important in early life, but that's under particular conditions. But I absolutely appreciate that probably other microbes will basically utilize those short chain fatty acids from an infection perspective. So it isn't just a case of going, yes, it's good, or yes, it's bad. It's probably context dependent as well. But I think lactate is one of the really interesting ones at the moment, and what that might mean. So yeah, thanks for bringing that
up. My other question is, I don't remember, I think it's the pill study or something, where you're moving beyond the gut microbiome. So obviously I think the gut microbiome is like stool, which is like the gut is interesting to work with because it's very high biomass. So I think you're also doing some skin swabs, I think. So one of the major complications of dealing with like something which is extremely low biomass, like for example, aerosols, when you do your sequencing, you collect your samples, right? How do you actually know that what you collected is actually that? Like, is it not just DNA background semi-control? So maybe my question is, what kind of controls, sampling controls are you using when you're doing the swabs? And how then, like, which method are you then going to use after you collect the samples and your controls? Are you just doing the normal beta, whatever, or you do use dequantum?
No, it's an incredibly important point about the challenges working with low biomass samples. And I guess if you're working with TB, then you know what those challenges are. So actually, Ileana, as part of the methods development paper, she put all of the negative and positive controls in there as well. So for each of our runs, we have a mock community that goes on there. We have our negative controls that run through the kit as well to make sure we've not got anything. And then we'll basically have a look at them to see potentially so we don't just do it on diversity because that's pointless. You need to actually look to see what's there. And also this is why looking at it through a microbiology lens is important, because you can look at it and going, well, that's a contaminant. Like, you know that immediately. But obviously there are some different tools that you are able to basically put that out. And of course, we're doing different batches. You can get batch effects from sequencing as well. So that's why we've got certain samples that we've basically stored, and we put the same sample in every single sequencing run as well. So we were lucky enough when we set up Pearl that there was lots of other brilliant work done by other people in terms of development stuff that we think we've been able to kind of control as much as we can in terms of making positive and negative controls to make sure that what we're seeing is actually what's there rather than just what's contamination. So absolutely, it's incredibly important.
Okay, yeah, sure.
Thank you.
Yeah, thank you. We just have one question online. Yeah, and thank you. So the question online was or maybe if you lend me, it's here. Yeah. Yeah. So thanks for the thank you very much for the fascinating lecture. What are the odds that the mother pass microbes that maybe read because I don't have my glasses? Yeah. Sorry.
And no, it's fine. And I don't need glasses yet. Yeah. Well, I think everyone can probably kind of read it and online as well. So, which is most likely maybe. Oh, interesting question. So basically, this is if mum's So if mum has a particular diet, which obviously influences her microbes, and those microbes get passed on to baby, does that mean that the baby then wants to eat the same stuff as mum does, because her microbes are probably the ones that like those particular foods? And that's a really, really interesting question. There's a lot of functional redundancy in the microbiome. So there's lots of microbes that can break down lots of different components. Some microbes that are uber specific. That's why shotgun or metabolomics, proteomics, and kind of functional studies are really important to get to that level of detail as well. But actually, it's a really good point about sustainably introduced probiotics later in life. One thing that for Bambi's, for example, and for the probiotics we gave, the reason that they worked, and obviously that was part of the data that I showed, was because it was almost a symbiotic approach. So that is a pro plus prebiotic. So giving a probiotic and then giving its favorite food to sustain it. And that's really, really important. So that's probably also the case also maybe in later life as well. So for example, some of these next generation like probiotics, like, I don't know, Faecalibacterium prasenciae or Akkermansia, for example, like Faecalibacterium probably needs a lot of fiber to maintain in the diet as well. And I've got colleagues that have done beautiful work on this in terms of looking at the adults. So it will not just be a case of probably giving a bug or a collection of bugs and hoping for the best. You're almost definitely going to have to tweak the diet to make sure that that maintains as well. But it's an interesting question about how the diet might influence the baby. And actually, we do have diet information what the baby's eating, so we could potentially actually answer that question. So we'll see how that goes. I always joke about that to say that, you know, my microbes like particular things, and that's why, like, Lindsay, you should eat more of this, when I shouldn't probably be eating more of it, but they're telling me to do it. But anyway, on that weirdo note, I will stop talking.
Okay, thank you so much. Yeah, we really appreciate it. Yeah, and I hope you enjoy all the questions. That's one of the whole ideas to enter in a discussion. Yeah, so just 2 quick notes. Yeah. We also value a lot public communication of science. Yeah, and we invest a lot of time on that. As a side event from the ISME, you say ISME, yeah. It is on the 27th of August. Yeah, we're gonna have a very large event for a few hundred. We have 200 or 300 high school students. Yeah. Which we're going to call Climate Change and Microbes, and that's going to be at the Kirstenbosch Gardens. Yeah. So anyone that wants to participate on that, get in contact. Yeah. Not only it will be fantastic, yeah, because we try to engage a lot with the general public, but it's very likely that all the flowers, the spring flowers, is going to be in one, which is one of the most beautiful botanic gardens in the world. I don't know if you have been to the Kirstenbosch Botanic Garden.
Last time I was here, yeah.
It's amazing, yeah. It's even worth it to bunk a session of the conference to go there. Yeah. So.
But not my session.
Not her session, which is Monday.
Monday,
at what time?
And we think 11. So we've got a morning session and an afternoon session as well.
Okay, so we're gonna be checking. I'm gonna be there as a police, yeah, to Bank on Monday at 11. I think that would be great to continue the discussion. And then also just to tell, next week on the 25th of September, yeah, we're also gonna continue with another external presenter on the microbiome, yeah. No, sorry, 27th of August, yeah. In the other week on Wednesday is going to be Carrie Kuhn from University of Wisconsin Medicine, yeah, and she will be talking about the microbiome in